Okay, so we're starting off here with the cardiac output, and this is the fourth objective in the cardiovascular system, basically understanding how everything is regulated and controlled. When we speak about regulation and control in the cardiovascular unit, that is going to mean the autonomic nervous system, right? Remember, autonomic nervous system controls smooth muscle, cardiac muscle, adipose tissue, and glands. So our formula for cardiac output is stroke volume times heart rate. And so this is a rate of the volume of blood that is pumped by each ventricle per minute. So it's going to be a product of whatever the stroke volume is. And we should remember, stroke volume is the actual volume of blood that's ejected. And if we multiply that by the heart rate, we should get the cardiac output. For an average cardiac output in a healthy male or female adult, it's about 5 liters per minute at rest. This can vary quite a bit depending on pathology, depending on disease state, depending on if you're exercising, this may be a little bit more, and other things like that. But average about 5 liters per minute. Now, if we look at our entire average blood volume, it's also about 5.5 liters. So we can deduce from this that every minute our heart pumps our entire blood volume out and around the body. So for every minute that passes, our heart is circulating our entire blood volume. So that really speaks to how much the heart is working and how important the cardiac output is for sustaining tissues. Let's talk about some factors that affect the cardiac output. So that's what this entire packet will be kind of going into in more detail. The two factors that affect cardiac output, so it's going to be from our formula, heart rate and stroke volume. And then we're going to spend some time looking at the factors that affect heart rate independently and spend some time looking at the factors that affect stroke volume independently. And so those factors will also affect cardiac output by affecting these two variables. So the heart rate and stroke volume are going to be regulated both intrinsically and extrinsically. And intrinsic versus extrinsic is something we're going to continue to see as we look at each system. We'll see this in the cardiovascular system, the pulmonary system, the kidneys. When we say that something is intrinsically regulated, it is regulated in and of itself. So this is a form of auto-regulation. So things happening in the heart regulate the heart's rate and rhythm. Whereas extrinsic, we're speaking about hormones, we're speaking about neural input. So things that are coming outside the heart that are influencing the rate and rhythm as well. So let's talk about some of those other factors or those independent factors. So here's a schematic that's showing we should remember this side of the screen. This is what the breakdown of the autonomic nervous system look like. And we can see here some of the preganglionic neurons that are innervating the heart. And so these are the sympathetic nerves, right? So we've got the preganglionic and then the postganglionic. This is the sympathetic chain, which we should be very familiar with at this point. And so we've got these nerves that are going to several places in the heart. We've got the SA nodal cells, the AV nodal cells, but also the myocardium, so the actual contractile cells of the heart muscle. Whereas for the parasympathetic system, we've got neurons coming from the brainstem. Remember, this is where our parasympathetic fibers arise, but they only innervate the SA and AV nodal cells. There is no myocardial innervation from the parasympathetic system. So just to kind of summarize here, the parasympathetic system via the vagus nerve affects the heart rate by affecting the SA nodal and AV nodal cells, but the sympathetic system affects the SA and AV nodal cells as well as the myocardial contractile cells. So that's going to be something we want to bear in mind as we continue to speak about the factors that affect each of these two variables, stroke volume and heart rate. So heart rate is determined by the SA node firing frequency. So we should remember the SA node as that group of autorhythmic cells located in the right atrium, the wall of the right atrium. And so the SA node has an intrinsic firing rate of about 100 per minute. And if we did not have any parasympathetic influence, our heart rate would be 100 beats per minute. But we know that in a normal, healthy person, the heart rate is actually about 75 beats per minute. And that is because of the parasympathetic influence that is slightly more predominant at rest, right? We talked about this in the autonomic nervous system. So although if you took an SA nodal cell and you put it in a beaker or in a petri dish on its own, it would generate or it would fire action potentials 100 per minute, but under parasympathetic influence, that is slowed down to about 75 beats per minute. If we're in an excited state, we know the sympathetic system takes over and the heart rate can increase. Whereas if we're in more of a rested, digested state, the parasympathetic will take over and the heart rate will be drastically reduced. Let's talk about the neural input to affecting the heart rate. So we know that if we increase the sympathetic system or sympathetic influence, nerves or epinephrine, so epinephrine either coming from the medulla, which goes via the bloodstream, or epinephrine that's being transmitted at the ganglia or at the synapse, which is in the form of a neurotransmitter. Both of these forms of this epinephrine will activate beta-1 receptors of the SA node. So remember, beta-1 receptors are the adrenergic receptors that are found in the heart. That will then stimulate increased opening in our sodium and calcium channels, which we know are depolarizing events, and that would create an increased spontaneous depolarization, which would make it really easy to fire action potentials, and that will increase the heart rate. So this is how we go from increased sympathetic activity to increased heart rate. And so here's what's actually happening in terms of the cellular events or the second messenger, the signaling cascade. So we've got norepinephrine or epinephrine, either as a hormone or as a neurotransmitter binding to this beta-1 receptor. It's coupled to a G-stimulatory protein. We know that's going to activate adenine cyclase, turn on our second messenger. We've got protein kinase that will then create increased opening of not only our sodium channels, which we call funny channels, but also our calcium channels. And if you remember from the previous packet, so the electrical activity of the heart, you should have an understanding of the cellular events that are important in bringing about an action potential in the SA nodal cells, as well as the events that take place in the contractile cell. And we know that these two channels must be open in order for these cells to fire action potentials. So if we are creating cellular events that increases the opening of these channels, it's going to be really easy to fire action potentials or spontaneously depolarize, and that is going to make the heart rate a little faster, okay? So this is, again, how we go from sympathetic activity to increase in the heart rate. Let's talk about how we go from parasympathetic activity to decrease in the heart rate. So we've got increased parasympathetic activity. Let's say we're resting, we're relaxing, the vagus nerve begins to fire. That will send out neurotransmitter, which will bind to muscarinic cholinergic receptors on the SA node, which will increase the opening state of different channels. So potassium channels, but also it's going to increase the closed state of calcium channels. And we know from previous knowledge that opening potassium channels is a hyperpolarizing event. And obviously, if we close calcium channels, that is also a hyperpolarizing event, as opposed to a depolarizing event. That is going to decrease the rate of spontaneous depolarization or increase the rate of hyperpolarization, and that will bring about a decrease in the heart rate. So this is how we do the opposite. We go from a parasympathetic influence, which results in a decrease in the heart rate on the SA nodal cells. So here's a schematic that shows that sequence of events. We've got our T-type calcium channels. We've got, if they're closed, calcium cannot enter, so we're kind of making it hard for a depolarizing event to take place. Likewise, we have potassium channels that are open, and they're going to be pushing potassium out, which will also make it hard for a depolarizing event or stimulate a hyperpolarizing event. Those are the same general actions, and that will make it really hard to depolarize this cell or really hard to fire an action potential. And so we're going to significantly slow down the heart rate as a result of this event. And all of that starts with acetylcholine binding to this muscarinic cholinergic receptor. And so we know these are the events that take place at the neuroeffector junction on the parasympathetic side. Okay. So here's a graphical representation of that relationship. So we've got three curves here. We've got the membrane potential on the y-axis here. We know that resting membrane is -70, threshold is about -50, and then we've got time on the x-axis. So under the influence of both sympathetic and parasympathetic, so at rest, we see the rate of SA nodal firing showing this here in gray. So the gray curve is showing what's happening at rest. So during this time period, we can fire 2 action potentials at rest. We also see the curve in green showing us what's happening under the sympathetic influence. And so we see that during that exact same time period, if we have increased sympathetic activity, we actually fire 3 action potentials instead of 2. And then alternatively, we have the parasympathetic curve in blue. And again, during that exact same time frame, under the parasympathetic dominance, we can only fire 1 action potential. So what we basically see is that sympathetic influence speeds up the heart rate. We can fire 3 potentials in the same time frame, and then parasympathetic slows it down, where we only fire 1 action potential in that exact same time frame. Let's talk about some of the hormonal controls. So when we speak about hormones, epinephrine is the main one, and it's going to have the exact same effects as the sympathetic nervous system. Remember, epinephrine can either be in the form of a neurotransmitter that is binding, or it can be a hormone that's coming from the medulla. So it's going to have the exact same effect as what we just described in the sympathetic nervous system. It's going to increase the action potential frequency of the SA node, which will increase the rate of action potentials in the cardiac muscle. Glucagon is another hormone that actually influences the heart rate. It's going to increase the heart rate. The mechanisms here are not exactly clear, but we do want to establish that glucagon is another hormone that does influence heart rate as well. And then speaking here briefly, we're going to look at how this is all integrated. Again, this should be very familiar to us. These sympathetic neurons are going to raise the heart rate. Parasympathetic neurons are going to lower the heart rate. And remember that comparative secrecy, the fact that both of these influences are acting on the SA node at rest. That's what keeps the heart rate kind of in the middle there at about 75 beats per minute.